Hello everyone, today we're going to be learning about protein synthesis. This is part of IAL Biology. It belongs to Unit 1 of the syllabus. Throughout the lesson, I'll be using my textbook. It's called the Complete Notes for AS Biology. This book has been around for 8 years and I keep updating it every year. In addition to the textbook, I'll be using also my classified exam papers. So these are questions taken from past exam papers that belong to every topic of the syllabus. So today I'll be using the questions that belong to the protein synthesis. I want to show you a quick look at what we're going to be doing today. So it's chapter 9. It's called protein synthesis from unit 1. Let's take a quick look at the notes. So as you can see here, the notes are based on the exam paper. So you can see here, that's a question about a comparison between DNA and RNA. So basically what I've done here, I took the marking points from the exam papers and I put them into a nice table like this. So it makes it very easy for us to learn and it makes it very simple for us to write the answers for exam papers. Another question is here, it talks about the process of transcription. That's today's lesson. And as you can see, I put them in steps and I put the marking points, the examiner's marking point, I put them in orange. I put the definition here, like when you see this sign, this means it's like a definition you have to memorize by heart, and the key facts in green, and every new term is going to be in. So here I have a quick overview of today's lesson. So you can see I have the double helix shown here. The double helix is the DNA, obviously. So we have a section from DNA shown here in the diagram. This section is what we call a gene. So basically what we're going to do today, we're going to take a part or a copy of the gene for any trait of ours. It could be for an enzyme, it could be for an eye color, skin color, whatever. And what am I going to do here? I'm going to make a copy of this gene because the DNA would never exit the nucleus. The DNA is very precious. We can't afford to take the DNA out of the nucleus. What we're going to do here, we're going to make a copy of that DNA. We call this process of copying of DNA, we're going to call it transcription. All right. And when we transcribe the DNA, or we make a copy of the DNA, we make something called the messenger RNA. So the messenger RNA is like single-stranded copy of one of the DNA strands. So that's the mRNA here. And what am I going to do with this mRNA? We're going to take this mRNA out of the nucleus. So this part, the transcription part, takes place entirely inside the nucleus. And what are we going to do now? We're going to take it out and we're going to go to the cytoplasm. Particularly, we're going to go to the ribosomes. On the ribosomes, we're going to translate that code into what we call a protein or a polypeptide. So here I have a bunch of amino acids joined together, attached together by peptide bonds. And that's what we call a polypeptide or a protein when it folds on itself. So that second part that took place in the cytoplasm, that's what we're going to call translation. Then this polypeptide would fold, take a specific shape, and it becomes a protein. This protein could be any trait, as I said. It could be your eye pigment or hemoglobin or whatever. Sometimes it's a bit difficult to understand how or what does each step of protein synthesis mean. So here I have a quick analogy to make it easy for us to understand. So first thing is the genetic code. Now the genetic code is stored inside our DNA. Now the DNA, as I said earlier, is found exclusively inside the nucleus. So the code itself is going to be copied. So I'm not going to copy, as I said, I'm not going to copy the whole DNA. I'm just going to copy a section. All right. So think of the DNA as a book of recipes, and we're only taking one of these recipes, and we're only going to copy one of these recipes. All right. So what are we going to call this copy of one of the recipes? We're going to call it the messenger RNA. All right. So messenger RNA in this analogy represents one photocopy of the recipes in that large book of recipes we call the DNA. What we're going to do now, we're going to take that messenger RNA and we're going to cook that recipe. So basically, we're going to turn a code into something physical. And to do this, we're going to take that recipe into the kitchen. So in this case, the kitchen is represented by the ribosome. The ribosome is where we're going to cook that recipe, right? And then once we cook the recipe, we're going to turn it into something useful, right? Like an enzyme, for instance. So that would be the final product of this process. So as you can see here in this analogy, we turn the code into something physical, useful, that we could actually make use of it. I need to stress on something that is important for us to understand here is the fact that we do not transcribe the entire DNA molecule, right? We're only going to transcribe a section of the DNA. That's what we called here, shown in green, that's what we call a gene. So we're only going to make a copy of a gene. Now, something also that is just as important is that we're not going to use both strands of DNA. You know that DNA is double-stranded. So we're only going to use one of those DNA strands, right? So DNA comes in two strands. One we're going to call the sense strand, just the name. And the other strand, we call it the antisense strand. Now, this antisense strand we also call the template strand. The reason we call it the template strand because that's where we get the information for in order to do the transcription or to make the messenger RNA. So while I'm transcribing, I'm only going to use this antisense or template strand, and I'm only going to copy this part we call a gene for me to make that messenger RNA. Throughout this lesson, I'll be solving some short multiple choice exam questions, and I will also be solving long written exam questions. So whenever you see this sign, the pause sign, just pause your screen and try to solve this question and any other question. Now, this is a quite simple question. They just gave you a strand from messenger RNA, and they wanted you to find the opposite strand on DNA, right? So you can see here it's a mRNA language or RNA language in general because you have mononucleotide uracil. But you need now to do what? You need to convert it to the DNA language, the one that contains thymine. So the first thing I would do here is just to cancel out option C because it has uracil. There shouldn't be any uracil in any of those answers. And I need to find the opposite pairs of nucleotides. So I'm just going to check the first four. So I have A, it should be a T opposite. U, it should be what? What should be the opposite pair or opposite base to U? It should be A actually. So for that reason, the only correct answer here is going to be answer A. So you can just make sure G with C and G again with C. So now that we have a good background about the process of transcription, we're going to go into details. I like to divide this process into three different small stages. The very first stage here is known as the unzipping or unwinding. So unzipping is an actual scientific word you can write in the exam. You can also write unwind. What does it mean in general? It means that I'm going to take that DNA molecule and I'm just going to unzip it. I'm just going to take away those DNA strand. I'm going to pull them away from one another. All right. So we said earlier that we have two strands, one we call the sense strand and one we call the antisense strand. So what am I going to do here? I'm going to just break away those strands one from the other, right? So here I have the two strands being separated. But how do we actually separate those strands? Those strands are joined by means of some weak bonds. So I have like weak hydrogen bonds, just like those you have in water, between the opposite pairs on DNA. What am I going to do here? We're just going to break those weak hydrogen bonds between the opposite DNA strands. Now I have that, we call it the antisense or the template DNA strands exposed. So what's going to happen? You have those bases, the RNA bases are always floating inside the nucleus. So it becomes easy for a uracil to pair up with an adenine here, or an adenine to pair up with a thymine, C with G, G with C. And as you can see now, we're starting to form what we call the messenger RNA. But that's still not the whole story. So what we've done so far, we unzipped, we did the pairing up of those bases, the RNA bases with the DNA strand. Still, I need one last important step. And that means what? That is the joining of the adjacent bases. So what am I going to do here now? We'll take a look at those bases. As you can see, they're only weakly bonded to the DNA strand. So what we're going to do next? Next is that we're going to make strong covalent bonds between those adjacent bases. Who's going to make those bonds? It's going to be an enzyme. This enzyme is called RNA polymerase. So RNA polymerase will catalyze a chemical reaction. We call that chemical reaction whenever we join bases or any molecules in biology. We call it condensation reaction. So we're going to do what? We're going to make some strong bonds. We call them phosphodiester bonds between those adjacent bases. Notice that I always stress on the word adjacent. Why do I have to write this in my answers? Because if I just say join bases, it doesn't make sense, because this could be any bases, like opposite bases, right? So that wouldn't make sense. So that's why when you write in your answer, you should really say the term or the word adjacent, so that they would understand, whoever's marking your paper, they would understand that you're talking about bases that are right next to each other on the messenger RNA. What I'm going to do now is want to link up transcription to translation. Transcription is the first stage of protein synthesis. I'm going to link it up with translation. So let's start with transcription. So as you can see, I'm starting here with a nucleus. That's where the initial part of transcription happens. That's when we unzip the DNA. We're not going to unzip the entire DNA because it's too big. We're only going to unzip a section. That's what we call a gene, right? So that's the unzipping part. Next part of transcription is we're going to do what? We're going to just pair up some RNA mononucleotide with those of the DNA. So as you can see here, I have a section, a gene here being exposed, and those are the RNA mononucleotide. What am I going to do here? I'm just going to take those bases, the RNA bases, and I will link them up with those of DNA. So you can see here that A goes and pairs up with a T, A with T, U with A, right? Because we're writing in the RNA language here, C with G, G with C, and that is not good enough because we still have to link up those adjacent RNA bases. So we have that RNA polymerase enzyme to do the joining part. So we're going to just link them up here by the strong phosphodiester bond. And the end product is going to be called here, we're going to call it the messenger RNA. So once again, let's take a quick look on how this thing happened. So that's the enzyme. That's the RNA polymerase doing its job here. It's running along the DNA, unzipping. And while it's unzipping, look what's happening. We're actually making the messenger RNA shown here in red. So you can see it's forming. And once this messenger RNA has been formed, it would soon exit the nucleus. How will that messenger RNA exit the nucleus? It's going to exit from one of those pores. You see those pores here inside the nucleus? So those are known as the nuclear pores. And that's how will the messenger RNA get out from the nucleus into the cytoplasm, and it's going to go and join the ribosome there. So as you can see, that's the end product of transcription, right? Made in the nucleus, messenger, and it joins the ribosome. That's where the translation part is going to start. So basically, translation starts when transcription ends. Here's a quick summary of the process of transcription. You can pause your screen and try to put those events in the right order. So to start with, I'm going to pick the first event and put it first. So the first event here is going to be the unwinding of DNA. So I'm going to take option B here and put it at the beginning because that one is unzipping of DNA. Following this, I'm going to take the pairing up part, so taking the the RNA bases and pairing them up with the DNA bases. Following this, I'm going to use the enzyme RNA polymerase to join the adjacent RNA bases, right? This means what? This means I've made the messenger RNA and it's ready to exit the nucleus. So this is like the ideal way of writing the perfect answer for transcription. I want to practice now this part that we've already done, transcription. So here's an exam question taken from past paper. It says, describe how a molecule of messenger RNA is made during transcription. The question is worth 4 marks. And as you can see, I've written more points than it's required. That would be very helpful to do in the exam. Like if the question comes for 4 marks, try to write 5 points because sometimes one of the points may not be part of the mark scheme. So that will help you to secure those 4 points of. So let's start by filling in the blanks. It says that the first DNA double helix, what is going to happen first? First, we're going to unzip or unwind, both are acceptable. Break in the hydrogen bonds, the existing hydrogen bonds, the DNA molecule. Then RNA nucleotides will line up opposite to the, what we call it, the template or the antisense strand, according to what we call the base pairing rule. Here is important to include the rule so they will understand that you realize it's A with U, not A with thymine or T. Then the next point, I'm going to say here phosphodiester bonds form between the adjacent RNA nucleotides. We're going to name the chemical reaction here. We're going to call it through a condensation reaction, which is catalyzed by the enzyme, the only enzyme we took today. We call it RNA polymerase. Following this, the mRNA, the messenger RNA, detaches from the nucleus through what is going to exit through a pore in the nucleus. We call that a nuclear pore. We're now going to start translation. So we've already done with transcription, we solved questions about it, and messenger RNA is ready now to be translated into something physical, and that is the protein that we're interested in. Translation looks more or less something like this. So as you can see here, that's the messenger RNA, the one that we've been working on so far in transcription, shown here in pink. It's going to go out of the nucleus, attaches to the ribosome in the cytoplasm, and there we're going to start building up the polypeptide. So you can see here, some other RNA molecule comes along. We call them the transfer RNA. We're going to talk about them in just a little while. And these start to dump the amino acids one after another until we make a polypeptide chain. Now, this polypeptide chain will fold on itself. It's not going to be called protein yet, so it's just going to have to fold on itself, and it would turn into a protein like hemoglobin, for instance. Now, let's talk about RNA in general. Now, RNA comes in three forms. One we call messenger RNA. That's the one we've been talking about so far today. And there's another one called tRNA, and a third one called rRNA. We're not going to talk about ribosomal RNA or RNA today because it's not really needed, but we're more concerned about those two RNAs. The messenger, that's the one that takes the message from the nucleus all the way to the ribosome, and the transfer RNA. Now, I always think of transfer RNA as a builder. Basically, it carries around what we call amino acids. So there's an amino acid always attached to the transfer RNA, I should say. And it basically does what? It basically starts to build the protein or the polypeptide chain. In real time, transfer RNA looks something like this. As you can see, it's not like really single-stranded. It's made of one strand of RNA, I should say, but this strand of RNA is looped on itself, right? So you can't really say that transfer RNA is double-stranded, but you can always say that it is single-stranded with some folding. So it folds on itself, and it has this, we call it clover-shaped structure. Now, as you can see, the looping and the folding of that transfer RNA occurs because of some pairing. So you see here, I have some bases, RNA bases that are paired up with each other. Like you have an A and U, C with G, they're paired up and they turn this RNA into that specific shape. And always in biology, the structure or the shape has to do with the function. So here on the transfer RNA, I have two parts. One part here on top, that's where the amino acids are attached. So always the amino acids, like you have an amino acid here, they'll be attached at this top part of the transfer RNA. And there at the bottom, you have three bases. Those three bases are very important. We call them the anticodons. So what's an anticodon? An anticodon is simply three bases. It could be like AUG or ACG, three RNA bases. And this anticodon is simply to pair up with some three bases on the messenger RNA. So like every three bases on messenger RNA, they'll pair up with three bases on the tRNA. So those, we call them the anticodon, if they're on the tRNA, anticodon. And those three bases here on the messenger, they are called the codons, right? It's easy to remember if the T here corresponds to the tRNA, right? Now, what's the point of this codon-anticodon pairing up that we have between those two types of RNA? Now, think about it this way. Remember, we have some amino acid that is always attached here on the top end of the tRNA, right? So this amino acid, so let's say this is amino acid, in my case, that's amino acid number 3. This will correspond to some triplet that is found here on the tRNA itself. and it would have to follow or has to correspond to the triplet you have on the messenger. So basically, the codon on messenger RNA determines what's the anticodon on tRNA, and it determines what the identity of the amino acid you have there on the other end of the tRNA, right? So let's try another one. If I take another triplet here, if I take like another triplet here on the messenger, all right? And let's say I'm taking the second triplet here, right? So 1, 2, 3, 1, 2, 3. So that triplet would be different from the first one, probably is different. And it would call for a different tRNA. So another tRNA will come along, and this other tRNA will carry some different amino acids. So as you can see here, whatever the triplet, whatever the codon you have on the messenger, it would basically attract a different tRNA, and each tRNA comes with different amino acid on one end and a different anticodon. Now that we have a good background about the types of RNA, let's try to write a good answer about translation. Like you've been asked to write about the process of protein synthesis in the cytoplasm, or they simply ask you to write about translation. How are you going to write a good answer? So as you can see here, I am in the cytoplasm on the ribosome. The ribosome aligns the messenger RNA in a way, making it exposed for those tRNA molecule. And as we said earlier, every tRNA comes with two parts. You have the amino acid part, where the amino acid is attached, and you have this triplet at the bottom that we call the anticodon, right? So the first triplet on the messenger RNA is what we call the start triplet, right? You have to remember that start triplet because it's always the same. We call it the AUG triplet. So that's always the first triplet on the messenger RNA. Now, AUG would correspond to what? It would pair up always with UAC. So that means that I'm going to put first amino acid here at the beginning. And then I have another triplet here, right? The codon, the second codon is called UGU. You can see it here. So now this will call for the second tRNA and the second amino acid. So there we go. So I have ACA that will pair up with the other triplet. So as you can see, what am I going to do here? I am basically putting one amino acid after another. And every time I have like two amino acid adjacent, like this, what's going to happen? The ribosome will catalyze a reaction, condensation reaction again, same as the one we saw earlier today. And that condensation reaction will form a bond between amino acid. We call that bond between amino acid, we call it a peptide bond. So that's we're forming a peptide bond through a condensation reaction. All right. So now that we've joined the first two amino acids, we're going to do more of this. So we're just going to bring some other tRNAs, some other amino acids. And as you can see, the polypeptide chain is getting longer. So those tRNA will soon exit. They will no longer need it. And you will be left with what? You'll be left with a polypeptide chain. So like the last triplet, will be third, or the last triplet here, the last codon. That's what we usually call a stop codon, called stop codon. So we have a start here at the beginning. We have a stop at the end. That will signal the end of the process of translation. So let's summarize what we've said here, what we've written about translation. So the process starts like what? The process starts with what you call the messenger RNA joining the ribosome. Then we have the anticodons on tRNA pairing up with the codons on the messenger. Following this, we have adjacent amino acids joining together by what we call peptide bonds. So you can see I'm using more or less the same key terms that I've used during transcription. I've used the pairing up and the joining of adjacent. We don't have unzipping here. We have messenger joining the ribosome. So when you're writing about translation, try to use those three lines or the three key terms. It would help you to write a good answer. Here's a typical question about RNA. They're showing us a picture of transfer RNA, and they wanted us to identify this section. So they wanted you to basically mention what type of bonding you have between this part of RNA, tRNA, and this other part. And I told you earlier that tRNA loops on itself to form some special structures looking like this. And the way that this pairing up of bases here and bases on the other end, this occurs by means of hydrogen bonds. So the correct answer here is going to be hydrogen bonds, because simply you have like bases like A, C here, and you would have here U, G, or whatever bases. That's another question here. They're asking you for part X. They wanted you to write or find what is usually attached at this part called X. Now remember, this section is where we have the amino acids. Those amino acids are always attached here, somewhere here, and they're needed for the process of translation. They're not needed for transcription because transcription doesn't involve amino acid or doesn't even involve transfer RNA. Lastly, they're asking you here to identify part Z. Now, we said earlier that part Z is the one you would need for the anticodon. This is the triplet here is called the anticodon. And the anticodon pairs up with the messenger RNA, the codon and messenger RNA during translation. In one exam paper, they asked the students at that time to make a comparison between messenger RNA and tRNA. So here I have like an extensive list of differences and similarities between messenger RNA and tRNA. So let's start with the part where it says folded. Now remember, only tRNA is folded because of those loops where we have hydrogen bonds. As you can see here, hydrogen bonds are exclusive for the tRNA. Both tRNA and messenger RNA are made up of the same mononucleotides, right? A, U, C, and G. So I'm going to leave this in the middle because it's common. Codons, the triplets, these are the triplets found on the messenger. So that's exclusive for messenger, while the anticodons are on tRNA. Notice that the tRNA is only found in the cytoplasm, while messenger could be found both in cytoplasm or the nucleus, right? So here it says in the nucleus. Messenger RNA is the only one that could be found inside the nucleus. Here it says variable length. So this is like a marking point in the exam paper. What does it mean when I say variable length? Now notice, tRNA has a specific size and length and number of bases, while messenger could be shorter or longer depending on how long the gene is on the DNA or how long the polypeptide chain is. Now here it says translational only, or only applies for translation. That would be tRNA. But here it says transcription and translation. Here we're talking about the function. So that would be messenger RNA. Amino acid attached, only tRNA comes with a part here on the top where the amino acids are attached. Now, let's make a quick summary of translation. So you can see here, I've put the key phrases, the ones that we need to write about translation, and the process starts like this. So first thing is the messenger, shown here in purple, joining the ribosome. And as they join, we start the translation. So this is the start codon. And what we're going to do now, we're going to pair up the codons and the anticodons, the codons on messenger with the anticodons on the transfer by means of hydrogen bonds, right? So now we've dumped the first amino acid. Then we get the second amino acid. So you can see it here. And we're joining the first and second amino acids together. You can see here, we're joining them by means of what we call a peptide bond, right? So that's the first peptide bond. And then we're going to pair up another codon and messenger with another anticodon on the transfer, right? And we make more and more peptide bonds until we get near the end of the messenger. Like the last triplet here doesn't really have a corresponding tRNA. So this codon here on messenger, we call it the stop codon for a reason, because there isn't any tRNA that would pair up with it, and that will signal the end of translation. So now I have what? Now I have the polypeptide chain, and that will be now ready to fold and form a protein. There's a link in the description to my website. It's called chem-bio.info. So just click the link. It will take you to the AS Biology section of my website. So there you will find a full package of all exam papers, solved exam papers. You also have videos for every single lesson, just like this one, that would cover the entire syllabus. You have links to other subjects like AS Chemistry or A2 Biology, and you also have very recent exam papers for all of the subjects.